delighted today to introduce our keynote speaker, Dr. George Church. Dr. Church is a professor of genetics at Harvard Medical School and professor of health sciences and technology at Harvard University and the Massachusetts Institute of Technology. He is director of the U.S. Department of Energy Center of Bioenergy at Harvard and MIT, and director of the National Institutes of Health Center of Excellence in Genomics Science at Harvard, and also director of the Harvard Personal Genome Project. He's widely recognized for his innovative contributions to genomic science and his many pioneering contributions to chemistry and biomedicine. In 1984, he developed the first direct genomic sequencing method, which resulted in his first commercial genome sequence. He helped initiate the Human Genome Project in 1984 and the Personal Genome Project in 2005. He's co-authored more than 300 papers and 60 patents and co-founded 9 companies, and is considered a pioneer in the field of personal genomics and synthetic biology. Dr. Church graduated from Duke University in 2 years with bachelor degrees in chemistry and zoology. Yes, I said 2 years. He began graduate studies at Duke University, but then transferred to Harvard University, receiving a PhD in biochemistry and molecular biology in 1984. Please join me in welcoming Dr. George Church.
Thank you very much. It's wonderful to be here. And to see so many familiar faces. This is my conflict of interest slide. I know it's important that you take everything I say with a grain of salt henceforth. And we'll be talking about this story of my saga of reading and writing genomes and the students and postdocs that have helped really bring this technology down in cost and up in quality. This is almost an iconic plot now in that we deviated from the Moore's Law curve from computing, which is in green, with this blue line that suddenly changed slope in 2005, which reminds us that these are not laws, these are just trend lines, and they can change positively or negatively, but it's exponential, such that instead of 6 decades, which was a fairly aggressive goal a few years ago, turned out it was even more spectacular, which in 6 years, and it needs to keep going down. It's around a little under 1,000 now, maybe 800, and it has to keep going down for reasons that will become obvious in just a moment. And it's not just about cost; it's also about quality. So when I started in this field, we would actually have debates as to whether 1% error rate or a little less than that was too challenging and still get our $3 billion goal for a human genome. It's a very interesting difference now today. And we have technologies that have changed over the years as well, not just for reading human genomes, but for writing human and animal genomes. And the ultimate writing is homologous recombination, or HR. And one of the enemies of homologous recombination is non-homologous end joining, or NHEJ. And you can see all these different mechanisms here. The ultimates, the ones that really don't involve non-homologous end joining, are these recombinases, but they're rarely used. And the star often these days, at least for now, is CRISPR, which we'll hear more about in a moment. But in addition to this difference between homologous recombination and non-homologous end joining, there's this issue of off-target. And it's the sort of thing that people obsess a lot about when we talk about gene therapy, and I just wanted to reflect for one slide on what it is we're worried about, because this is not often explicitly stated. What happened was between 1996 and 2003, there were these 2 papers, the first one being a bit more positive than the second. And in between, what happened is patient 4 and patient 5 in this SCID study died due to integration of the gene therapy vector stimulating the LMO2 oncogene in two different integration sites, indicated here by P4 and P5, causing proliferation of mature T cells. So this is what we're trying to avoid. We're trying to avoid either stimulating oncogenes or inactivating tumor suppressors. So fast forward to today, and there are now 2,000 gene therapies in clinical trials, 2 of which have been approved outside the United States. And they tend to either use non-integrated vectors, although non-integrated is really a euphemism for 5% integration. And then there's gene editing, which is just beginning to make its way into clinical trials. In particular, there's a CCR5 very promising gene editing from Sangamo. And this is what we're worried about with gene editing, is that there's some hundreds, we don't know the exact number of human tumor suppressor genes and hundreds of oncogenes, and we can make nucleases or activation versions of the CRISPR-Cas9, more about this in a moment, but just take it for granted we have nucleases and activators, and the nucleases we want to avoid the tumor suppressor genes, the activators we want to avoid the hundreds of oncogenes. And if you do the math, you end up with, you'd like to have an error rate less than 2 times 10 to the 19th, 1 error in 2 times 10 to the 19th base pairs. And that sounds daunting. Hopefully you agree with me, that sounds daunting. The best that I've seen in the literature so far, and this has not really been confirmed in other laboratories, but I think it's probably true, is from Keith Young's lab, Shendar Zai, first author, where that's at least 3 times 10 to the 14th base pair error rate, meaning no error is observed in that number of base pairs. Okay. So what can we do to improve? So that's with CRISPR-Cas9 as it comes out of nature almost. Think of one of these as a CRISPR-Cas9 where you have a guide RNA. I know from experience I don't need to say much about this to this sophisticated audience, but you have a RNA-mediated recognition of the genome, and you can either have it the natural form, it makes a double-strand break. You can mutate it so it makes only 1 nick, or you can mutate it further so it makes no nicks. And then you can require the coincidence of 2 of these near one another, so that you, in order to get a full break, the break that will cause a mutation or stimulate homologous recombination, you need to have both of these near one another. So if one of them binds in the wrong place elsewhere in the genome, that is unlikely to cause an off-target deleterious effect. You need to have them both close to each other. And then there's a variation on this where you make a non-nicking form of the Cas9, but then add it to a dimeric FOK1 bacterial nuclease, which require, but both of these have in common, they require dimerization, have 2 20-mer, so 20 nucleotide recognition elements for the RNA, DNA triplex here. They have to be near each other and defined computationally to be rare or missing, except where you want it to be. Anyway, so here's some of the references on these ways of improving specificity, and the hope is that we're already so far beyond the 10 to the 14th that we're maybe at 10 to the 19th, but we're really getting, the challenge is measuring and documenting the error rate. Ultimately, it will be determined by preclinical animal trials looking for cancers and other off-target effects. So I just want to provoke and stimulate you with kind of some of the bleeding edge things that one can do with genome engineering. There's one class which is not CRISPR. It's sort of the world has come to this funny state where it's with and without CRISPR. So just briefly I'll mention how we can use recombinases. And then there's some various topics of which we really only will talk about one of these, but gene drives, xenotransplantation, aging reverse and de-extinction of arthropods. Just we can improve the specificity of recombinases, just as I mentioned that we improved the specificity of the nucleases. I'm just going to show the reference and not go into the details, but it has to do with this dimer binding interface is the critical things. There are two kinds of recombinases to keep straight here. One of them is a four-way junction where you're bringing together two pieces of double-stranded DNA. Another one is where synthetic oligonucleotides bind at the replication fork and pretend to be these Okazaki fragments. They pretend to be these little these short pieces of DNA on the lagging strand. And both of these have come together in the engineering of the first genomically recoded organism. Now, I'm not using recoding here as a metaphor, but literally the genetic code of these organisms, one of the 64 triplet codons has been changed genome-wide, every single instance in every gene, so that the organism has a new code. And the first thing you should ask about everything that I say and everything we all say is why are we doing this, right? It's not just how, but why. And we have four reasons in this case. One is we have many uses for non-standard amino acids, some of which have already been validated in human pharmaceuticals by Ambrx. We can use these non-standard amino acids to achieve genetic and metabolic isolation, which is unprecedented. We've shown that we can get better than 1 in a trillion background levels of isolation, and we can probably do better and will continue. That's 0 out of a trillion. We may already be better than that. And we get multivirus resistance. This was something that dawned on us partway through the project, and we realized that, and we have observed multivirus resistance. And it's an interesting reflection that we can be that these organisms can be resistant to all viruses in nature, even ones we had never studied before. This is in contrast to the normal, very difficult slog of finding ways of making cells resistant to viruses through, say, pharmaceuticals or deleting the receptors for the virus requires deep knowledge of the virus. This is just simply the virus doesn't find its familiar genetic code. And these were published in Science and Nature. The Nature one just came out. So now back to CRISPR and a particularly exotic thing that one can do with it. This is not a standard practice by any means. But it builds on something that occurs in nature, which is called a gene drive. I was first introduced to this as a graduate student, where Bernard Dujon, my mentor, along with Wally Gilbert, but Bernard had discovered a piece of DNA, a selfish piece of DNA that didn't jump around randomly like transposon. It would insert in a particular place in the ribosomal DNA of yeast mitochondria. And this became the first meganuclease, for those of you who appreciate the origins of our genome editing world. And that meganuclease was responsible for the homing and the selfish nature of the DNA. Now you can make this DNA a little less selfish by giving it a payload which is useful to humanity. And one of the payloads that we're working towards, and by we, I mean here Kevin Esfeld, who's a fellow, and Andy Smidler, who's a joint graduate student between my lab and Flaminia Cataruccia at the Harvard School of Public Health. The more altruistic payload we're looking for in this gene drive are genes that provide resistance to mosquitoes, resistance to malaria. Malaria, as you all probably know, is a horrible force of poverty in the world, as well as killing 600,000 people every year, and millions are deprived of valuable work hours and family time. This is the idea behind the gene drive is you have a cassette that carries the CRISPR nuclease and guide RNA, as well as the payload, and it is flanked by homology regions where the CRISPR will cut in the wild type, but not in its gene drive version. So there's a difference, a recoding essentially of an essential gene, such that it has this gene drive adjacent to it, and And it will only, whenever you make these two, instead of having 50% of the progeny inherit the gene drive, if it were just an ordinary piece of Mendelian DNA, it actually cleaves the wild type version and converts it to gene drive. And now they all have this highly desirable payload, which makes the mosquito resistant to malaria. This is we have Cas9 working in mosquitoes, as we do in almost every organism to which you can deliver DNA, every organism that's been tried by many labs. We do not yet have the gene drive working here. We have it working in yeast. And it's quite remarkable, actually, and another lab just reported it in Drosophila, which is, and the result is, in yeast, it's greater than 99% of the offspring, instead of 50%, get converted. And in Drosophila, it's somewhere between, it's about 97%. So this is extremely feasible, and in one season of mating, you get hundreds, each mating cycle you get hundreds of matings, and then a couple of weeks, and many cycles of these 2 weeks results in exponential growth throughout the population. And so we have cases of human malaria in the malaria belt. We also have, this is applicable to invasive species. There are, for example, rodents are responsible for the largest mass extinction on islands in recorded history, and many of these are now being removed and showing the feasibility of avoiding extinction and of using gene drives, at least on islands. Another slightly exotic application of the CRISPR nuclease and homologous recombination methodology is in humanizing animals, in particular pigs, that might be used for xenotransplantation and possibly testing other therapies. There's been great progress from a number of labs here. We're working with two of the long-term pioneers in this, Joe Tector and his team, and David Sachs and his team at MGH. And CRISPR is being used to change multiple genes, some of which have already been validated. But the most challenging one is the endogenous retroviruses, of which we count about 40 copies of active endogenous retroviruses. And these have been shown by other groups that they are capable of crossing over from pig cells to human cells in culture, and then going from human to human cells. So this is more than a hypothetical, but getting 40 genes converted, we and other labs have done 3 genes simultaneously with CRISPR, but now we're highly motivated to do 40 at once, which is challenging. But we can make organs in ways other than making them in animals, and we're pursuing some of these other ways. These include 3D printing, but most powerfully, developmental biology in cell culture. Cell culture has been fairly static for about 100 years, but now it's moving into these organ cultures, which are quite exciting. When we test hypotheses, we don't always have compelling evidence, even when we have large cohorts, and occasionally we'll have a cohort where we'll have one affected individual in the world. This is a case of a myostatin double null, where there was one well-documented case in the world of an infant with high musculature, and this has been now one way of repeating it in animals, 3 different animal models here, bovine, dog, and mouse. You actually don't need the large cohorts anymore. You're actually moving to causality, but you need to have a very sharp hypothesis and very convincing outcomes. But the alternative, which I was building up to before this slide, is doing this in animals. There are cases where the animal model is inadequate. For example, the cerebral cortical organoid has anatomical and physiological functionality not found in the mouse brain. It's found in the human brain and in this organoid. And our wonderful collaboration with people in the Wyss Institute, like Don Ingber's lab and Kit Parker's lab, where many different, about a dozen different complex tissue types, they're not quite organs, organoids, where you can reproduce many of the mechanical forces and the physiological significance of having an air, epithelium, endothelium, blood interfaces, for example, in the lungs. We just heard a talk a little while ago about using looking at cardiomyocytes, but Kit Parker has developed a very sophisticated contractile model where you're getting more than just a clump of cells, but you're getting very highly ordered structures that you'll see in just a moment. We did this in collaboration with Kit and with Bill Boo's lab. That Bill's providing the insight into this Barth syndrome cardiomyopathy, where a single G out of the 6 billion base pairs in the genome, single G on the X chromosome was thought to be deleted, and that be the causative allele for this. There are millions of differences between any two individuals, but this one seemed quite promising as a candidate. And the way we prove causality in this case is to use a pluripotent stem cell line. This is one of our favorites because it's fully sequence characterized. You'll see how important that is in a moment. And we have doxycycline-inducible Cas9, which is excisable by a piggyback system. So it's kind of a hit and run. You can make a clean genome that has just this one base pair change by this hit and run method. You can also, if you include donor DNA, you're getting homologous replacement of this one base pair. If you don't include the donor DNA, you get non-homologous end-joining, which can be either insertions or deletions or any combination. In this particular case, we characterized a genome that had an 8 base pair insertion. And this is what it looks like. We have the wild type, which is a mock CRISPR modification. And you can see this highly ordered sarcomere organization, repeating structure of contractile muscle. Remember, this didn't come from a patient. This came actually from my cells, I will confess. Taken from fibroblasts from my left arm, turned into stem cells, and then some stem cells into this highly ordered cardiac tissue. If we make that one base pair change to test causality, you can see the highly disordered structure and the great loss of contractile function. And then the non-homologous end joining, which was +8, similarly. And to complete the proof that this one base pair is causative, you can complement it in trans with a modified messenger RNA encoding just the TAS gene product, showing that it's this G in the TAS gene which is causing the disease. Now, I've glibly said along the way here that we changed one base pair out of 6 billion in the genome. Why can I say that with such confidence? And probably those of you who know me know that my postdoc, Luhan Yang, sequenced all the genomes we were talking about. So it is now considered routine for a postdoc, if they change one base pair, to sequence all 6 billion base pairs and make sure they didn't change anything else. And these are clonal, so we're not talking about a population of cells that are heterogeneous, which would happen if you apply CRISPR to a population of cells, which is another way of doing this. And in fact, she did it both ways, and this summarizes what happened. And it illustrates a very interesting lesson that where a small mistake gives us a very large lesson. So here he has a number of mismatches, and ideally, obviously your target has 0 mismatches, and there's one site on chromosome X at this position, and it's deletion of 1 G. And the frequency here is fairly high. CRISPR in some systems get us up to 80% frequency. And then we try to avoid anything with 1 mismatch or 2 mismatches. The algorithm, the computational algorithm, is a little bit more complex than that, but just rule of thumb, you avoid 1 and 2 mismatches elsewhere in the genome. So you scan the entire genome for a match to your guide RNA 20-mer, and you try to avoid those mismatches. And you typically can. Now, why didn't we in this case? This is the interesting point, because we didn't, because we allowed one with 2 mismatches, which happen to be on chromosome 5, it's a SNP. It's not present in the reference genome, which is the computer checked, quote, the reference genome, the one that many people use, and it didn't have this SNP. But my genome, which is the genome we should have had the computer look at, does have this SNP, and as a consequence got this huge off-target. So the lesson here is know your genome, and if you're going to be doing gene therapy either for research or for clinical use, there might be an advantage to using the correct genome as the reference sequence. And then if you do a large population of cells, you can see that there's some with an off-target of 3 or more, and these constitute a 5% background. In a clonal assay, we got 2 clones without any of these off-targets, but in a large population, you can see that it would have been 5%. So there are a variety of tools looking for off-target effects, and I mentioned earlier how low we want that to go. But there's also, it's important that we have these tools for testing hypotheses where we can introduce mutations. You don't necessarily have to have access to the patient cells in all cases, although it's very, very good. And one way of getting access to general stem cell tools or patient cells that is an open standard, it's now worldwide, is this Personal Genome Project. And I actually know that there's some people that participate in this project here in the room, like James Shirley, who's one of the pioneers in this. And it's been recognized now in a unique collaboration between the National Institutes of Science and Technology and the Food and Drug Administration. It's called genominabottle.org. And they searched the world for appropriate cells and DNA that they could use to make the standards. And they found that only the Personal Genome Project, personalgenomes.org, was suitable for this in properly consenting individuals for re-identification and commercial use. So they selected 8 trios that were intentionally diverse ancestry, and these are publicly available as cells from Coriell, stem cells and B cells. And what's important is not only do we have the cells available for human synthetic biology and gene therapy tests and causative analyses of disease, but we have their full genome and many, many other traits. It is not looking for a particular disease in all these individuals, but getting a large, or what's called big data of precision medicine about each of these individuals, shareable the way that a physician would see a big precision medicine data set in the near future. It is not sufficient to have a de-identified genome and a few facts about an individual. You really need, if you want to study precision medicine, you need to have these big data sets. So I urge you that if you're interested in volunteering, you know somebody that is, look at, just do a Google search on Personal Genome Project. Now that was an instance of a fairly rare disease. We're also interested in common diseases. And the most common disease, hopefully all of you know which one I'm talking about here, is if you look at age, you have this kind of skewed bell curve, and there's so few people who make it past 110 that that you really can't see it in this plot, but you'll see it more clearly in this following slide. It's on the order of 70 people, steady state, in the world at any given time. And here are some of them. And we have, in a small consortium, sequenced a significant fraction of these individuals. And I can hear a little snickering, and the point of this is not that you start up your smoking and drinking and sugar eating in order to survive. We think that they survived in spite of all of this. They have an environment similar to ours, but they have a very different reaction to it in terms of resistance to infection, cardiovascular decay, cancer, and so on. Now, here's an example of progress in this direction. Not just longevity, which is a very difficult assay to do for a graduate student who wants to do a human longevity assay. They have to wait 120 years to get their PhD. Aging reversal is a more suitable assay, and here's a particularly interesting one called heterochronic parabiosis that my wonderful colleague and collaborator Amy Wagers and others have done, where they will fuse the circulatory system of a young animal mouse and then use that to purify factors that might be causative in the rejuvenation of the older mouse in this heterochronic parabiosis paradigm. And one of these, one of hopefully many factors that have been isolated so far is called GDF11, and this can be shown by itself to be instrumental. A separate line of research on age-related decline, and we need to be careful that reversing age-related decline is not necessarily going to, of a particular biomarker does not necessarily reverse the decline itself. But here's something where we know there's all kinds of mitochondrial homeostasis loss, and in particular NAD drops between age 6 and 22 in mice by about twofold. And so this could be reversed by a small molecule that's very expensive, or we can target one of the regulatory transcription factors just upstream from this mitochondrial homeostasis called TFAM to make something that, rather than being a multiple time per day dose, can be a once in a lifetime dose, where we use CRISPR as an activator to activate just upstream from the transcription start site for the TFAM, which itself is a regulator. We can make artificial regulatory sites by just looking for potential CRISPR sites, and you can carpet bomb the region upstream from the transcription start site for TFAM, looking for one that is highly activating, using it by taking the null, the nuclease null Cas9, putting an activating domain on it. VP64 is one of the favorites. And you can see that one of the 4 sites we tried out has a big effect, and some are very tiny, barely noticeable, but you can get up to a 47-fold activation, which is more than adequate to get you return of your twofold, in fact, closer to 10-fold NAD impact. So you can dial it up and down in that range. But for some places, you might want to have a bigger than a 47-fold increase in a particular gene product. And so we persisted in improving the activation domain for this otherwise dead Cas9 or dCas9 by adding not just as a VP64 onto the C-terminus, but P65, which is an independent transcription activator, and RTA. The first letter of each of these is VP and R, and so it got the acronym VIPER, and it happens to be that this is the optimal arrangement of these 3. We tried many combinations of and many other activation domains, and this is the winning combination, where you can see up to a 20,000-fold impact of genes like Titin, which are so large that it's hard to deliver the gene itself in a gene therapy setting where you might have viral or non-viral delivery, because just the coding region of the gene, much less all the regulatory and intron part of the gene, the coding region alone is 100 kilobases, very large payload for a small virus. but you can deliver this, this smaller CRISPR activator and get 20,000-fold impact, which is a big improvement over the VP64, which is about 64. And you can do multiple of these at once. So you can do here 4 at once, getting so in the range of 400 to 3,700-fold impact. Okay, I just want to end on provoking some questions here. On how we analyze this, kind of coming full circle back to reading and writing genomes and epigenomes. We've been talking about how we manipulate the gene expression with a new method that we described in Science recently for in situ sequencing. And the challenge here is that many cells are, you can do assays on single cells, but many single cells are not spherical. For example, neurons are not. And the subcellular distribution of RNA and protein, as well as the multicellular context, is extremely important. So we developed this method over a 10-year period. Some of our methods go very quickly. This one took a while. Here are 2 cells where the nuclei are slightly brighter, and each of these dots is a single messenger RNA molecule which has been amplified. And in contrast to standard next-generation sequencing, where you might grind up a tissue or a cell and then randomly distribute its sequences onto a slide for the determination. In this case, you read them, you amplify them in situ, so you get the same kind of sequence analysis, either Illumina or solid ligation chemistry, which is fluorescent. You get the sequence, but you don't lose the 3-dimensional coordinates, so you get the subcellular and multicellular context of each of these dots, which is a single messenger RNA molecule, which has been amplified during the process of making cDNA. And you make a hydrogel, which is capable, which maintains a structural integrity, but has very easy flow in of reagents, so you can go through the cycles of sequencing over and over, and essentially get an infinite number of colors. Now, one of the ways we assess the utility and quality of this is whether we're getting good coverage all along all of the messenger RNAs. And here is a typical messenger RNA, but a rather long one, which is 8 kilobases long. Hard to get full-length cDNA, but we don't need it because we have all these short reads all the way along it. And you can see it's almost every position on the messenger RNA is represented. One that was particularly striking when we first did this experiment, sort of halfway through the experiment, which was a human fibroblast wound healing paradigm. We noticed exon 25 was completely unrepresented in this, and this was distressing. But then we continued the experiment anyway, despite the possible artifact, where you do the wound healing and change the media to epidermal growth factor, and then we found multiple reads in exon 25, and indeed exon 25 is a case of alternative splicing that has physiological significance. So, going forward, and in conclusion, we're extending this measurement of epigenetic changes, whether it's done in the context of developing causal analyses or developing organs for testing drugs. We want to be able to get very good epigenetic modification, even epigenetic therapies. And anyway, two of the ways, improving this fluorescent situ sequencing, we have 3 different ways of obtaining higher resolution. The first one is simply, we call it molecular stratification, but this means we can read multiple reads per pixel. You might think, oh, that's kind of weird. You know, usually if you try to read multiple sequences at once from 1 pixel in an image, the computer should get confused. But if you do them one at a time, you do one sequence and then you strip it away and then you do another sequence at that same position, you can get multiple sequences no matter what your resolution is from a single pixel, as you might get if you want to get multiple reads per RNA. And the one I'm most excited about just came out from Ed Boyden's group, with whom we've been collaborating for a year, called expansion microscopy. And rather than trying to improve the resolution of microscopy, for which the Nobel Prize was given, where you keep pulsing on or keep examining the same pixel over and over, maybe thousands of times, in order to get an accurate centroid of that. So you get a higher resolution than the light microscopy was thought to be capable due to the diffraction limit. But an alternative is just to expand the sample. So literally the same hydrogel that we use to embed for fluorescent situ sequencing, similar kinds of polymers have the property that when you change the ionic strength, they will swell up to 10-fold in each direction, X, Y, and Z. And now, rather than the extra time it takes to examine each pixel thousands of times in order to get an accurate centroid, you can now use conventional, say, confocal microscopy to go at 1,000 times faster, but still get incredible super resolution. So a factor of 10 in each dimension is a factor of 1,000 in terms of the reads. So I'll just leave you with those thoughts about how to do genome engineering, epigenomic engineering with transcriptional activators and repressors, and then finally how to analyze genomes and epigenomes in situ. Thank you. Questions?
Do you believe that everybody should get their genome sequenced, or just specific people that have like risks for diseases?
That's an excellent question. The standard practice until recently has been that you need to have a very good reason to get your genome. It would have to be a family history or unexplained so-called developmental odyssey for a child, genetic odyssey for a child with developmental delay. I think that is caving in as we get better and better data on background levels of these mutations in the population, so we know which, let's say, BRCA1 and 2 alleles are not impactful and do not require preventative surgery to prevent breast cancer. But there are many, many actionable alleles, predictive and medically actionable, that all of you are, as far as I know, susceptible. You don't know until you examine it. And so I think that if you're thinking about having children or there is adult onset diseases, it's getting to be cost effective, but it obviously has to be done with great care. But I think the simple answer is I personally believe, it's just an opinion, that we all should get our genome sequenced, and if we can't afford it, then we should be lobbying various people to make it affordable. Yes?
So what are the limitations of genome engineering? I mean, is it safe to use it like clinically yet?
So, right, so there is one genome editing clinical trial that I know of that's in phase 2. It's the CCR5 deletion mutation that Sangamo is doing to treat patients with HIV, so by deleting the HIV receptor. I think it may end up being the most specific pharmaceutical we've ever seen. You could basically go from target to therapy, and using these tricks for making it highly specific, you can have extraordinarily low off-target. All drugs have some kind of off-target. But we need to proceed carefully, as we do with all drugs, to first through animal models, and then and human cells ex vivo, and then finally into small phase 1 trials, and then scaling up. But I think you're going to be seeing this go from an idea of a technology 2 years ago to clinical phase 1 trials a year from now. So a total of 3 years, which is pretty good for a new technology.
Yeah, but was it like, is it yet applicable? Like, was it used?
So there are many preclinical animal trials that are looking very promising for a variety of human diseases, yes.
Thank you.
Is there any way to use gene therapy in like an antibacterial way, like to target a bacteria and have it kind of kill itself, I suppose?
So this has been proposed, and some experiments have been done. It has the challenge for both infectious disease and cancer is you're fighting a Darwinian battle where if you leave a few survivors that have survived because they're resistant either to the CRISPR uptake or whatever drug you're using, the uptake or the mechanism once it's inside the cell, those will proliferate and then you have a resistance. I think the better use of these, I mean, we should explore all avenues, but a very exciting possibility is preventative medicine, where rather than waiting until you have one of these exponential Darwinian losing battles, you create cells that are intrinsically don't have a receptor, for example, in the CCR5 I just mentioned, or you preventatively, genetic surgery, the equivalent of current surgical removal of organs at risk.
I think what you said kind of ties into my next question. You're probably asked this a lot, but do you think we'll ever get to like a Gattaca-esque thing where we can edit the genomes before people are born and maybe prevent diseases too?
As I recall, Gattaca was more about selection than about editing, but your point is well taken. I guess Gattaca wasn't entirely negative, but I hope that we can do better. I think we need to be thoughtful about what we want, not just for medicine, but for humanity, and to maintain ourselves. Do we want to commoditize and mechanize and medicalize our current diversity? And by diversity, I don't just mean the diversity represented in this room, but I mean neural diversity, for example, where there are people that are different, but make valuable contributions. And I'll raise my hand as one of those that was not exactly fitting in when I was young. So I think we have to be very careful to avoid getting rid of people that seem to be on the losing end of the genetic spectrum.
One final question. So what you're proposing is similar to like what 23andMe is saying with personalized testing and then personalized pharmaceuticals from the genomes?
Well, yeah, I think personalized pharmaceuticals or pharmacogenetics is one application of it. It's probably not the major one right now. I mean, the preconception, genetic diagnosis, genetic counseling, non-invasive prenatal testing is really taking off. There was almost none of it a couple of years ago. There'll probably be a few million in the next couple of years. And then newborns and then adult onset. It's not just about pharmacogenetics. I'm not even sure that that's the major application.
Thank you.
Yeah.
Dr. Church, if he would just speak to one more issue, and it's consistent with what the New England Science Symposium is about, and if you see his research and his science and how brilliant it is, but I also know he's equally committed to the scientists of tomorrow. And I'd like him to just briefly give your thoughts on possibilities for the future as we look out at an audience of people who want to make a difference.
Well, it's a great question. I actually just gave a talk at NSF, which I was tempted to give again today, but I tend not to give the same talk twice, on how we encourage, this was at NSF in DC, how we encourage the next generation to think out of the box and come up with transformative research, and for that matter, very solid, rigorous research as well. And how do we give subtle discouraging comments in various ways. There are many ways that the journalists, for example, can be discouraging by not getting the message right or distorting it. Your colleagues, and in fact ourselves, we tell ourselves very often these really transformative ideas come to probably everybody in this room, and they are dismissed within a few seconds before you actually even give it full respect that it deserves of rejecting it because you've got solid reason why it's not possible, speed of light or some fundamental principle of physics. So I think that's part of the future is creating a culture in which failure is okay as long as you do it quickly and move on, and where ideas that are radical are treated with respect and for a while and not undue levels. And safety is a very high priority. Safety for the individual, for respecting the humanity of each individual, and respect for the environment and for other people's opinions.
Please join me in thanking. Wonderful, wonderful presentation. I was sitting next to somebody who was writing down all these science articles and these nature articles and all of these other kind of things. So I think you have a lot of reading to do, is what I was thinking as you were writing down. I was just thrilled. And my next thought was, how can I get more students and postdocs into Dr. Church's labs? That was my other thing that I was thinking. We've had a wonderful morning, great presentations. We're going to turn to the next part of the program and lunch. There's 3 sets of individuals, so think about this. The oral and poster presenters are going to go to the rotunda on the 3rd floor. There is an elevator outside of the doors on this side, and if you walk to the other side of the building, there's another elevator. 3rd floor, oral and poster presenters. For all others and guests that are here, there's going to be lunch served in the Elements Cafe where breakfast was. Go outside, go walk right up the stairs, 1 flight of stairs. If you don't want to go up the stairs, take the elevator up 1 flight. And that's for general attendees. And for all of you who are here, who are part of the individuals who will be judging the posters and the judges from this morning, the oral presentations, on your folders, it will tell the specific room that you're supposed to be going to. And your lunch will be served in that room. So judges, going to the specific room, oral poster presenters to the 3rd floor, all others to the Elements Cafe. Following lunch is the poster presentations. They are amazing. Come back to your posters and be ready for people to ask you questions at your posters. And we'll see you this afternoon.